Hey everyone, welcome back. We are continuing our discussion on the integumentary system. We just finished talking about the epidermis. This is chapter 5, by the way, chapter 5, slide 18. We just finished talking about the epidermis. We're starting to talk about the next layer down deeper, the dermis. The dermis is divided into two layers, and unfortunately, one of these names is kind of misleading. It's divided into the papillary or papillary layer and the reticular layer. The papillary layer is called that. If you look down here at this diagram of skin, you can see that this papillary layer has bumps. The word papilla means bump. The word papilla means bump. So this is the bumpy layer, papillary layer. The papillary layer, if you recall, without even knowing this name, it's immediately deep to that epidermis. It's immediately deep to the keratinized stratified squamous epithelium. We know what this is made of. This is made of areolar connective tissue. It's made of areolar connective tissue. It's part of the cutaneous membrane. The reticular layer is called that not because it has reticular tissue. It's called that because it's a network of something. Got a network of blood vessels and nerves and collagen. I wish there was a different word for this, but that's what it's called, the reticular layer. What it's actually made of, let me just write that down. Network of collagen, blood vessels, nerves. Does not have reticular connective tissue. What it does have is dense, irregular connective tissue. It's dense, irregular. That's also part of the cutaneous membrane. So altogether, epidermis plus papillary layer plus reticular layer of the dermis, that is cutaneous membrane. Deep to the cutaneous membrane, that's the subcutaneous. Deep to the cutaneous membrane, that's the subcutaneous. Subcutaneous, below the cutaneous, also can be called hypodermis. It's below the dermis. It's not part of the cutaneous membrane, but together with the cutaneous membrane, that's what makes up skin. That's the thickness of skin. It's made of adipose, if you recall. The histology here on the right, you can see the papillary layer is roughly that. It's a very slight difference in the texture, the appearance of the papillary layer, which is areolar connective tissue, versus the reticular layer. Do you see the broader collagen? That indicates that it's dense, irregular. All of this is reticular layer. Deeper to this, if we keep going down, we would find adipose. But the reticular layer is much bigger. Papillary layer is narrow, thin. Reticular layer can be very big and extends down, and then we find the subcutaneous adipose. Let's draw this on our diagram that we were working on previously. Deep to the stratum basale, we have very light colored tissue. and it follows those bumps. This layer is the papillary layer. Deeper to that, we start to see collagen that's thicker. And darker. This is the reticular layer. Deep to that, which I'll only be able to show a little bit since I've used more real estate here for the other layers. Deep to that, we have large cells, large round cells that make up the subcutaneous. This is adipose tissue. You can call this the subcutaneous layer. You can call this the hypodermis. It's the same thing. This layer, to remind you, is made of adipose. The reticular layer is dense, irregular connective tissue. The papillary layer is areolar connective tissue. And then all the epidermis let's try that again. The entirety of the epidermis is keratinized, stratified, squamous epithelium. The papillary and reticular layer together make up the dermis. The epidermis and dermis together make up the cutaneous membrane. So you have the cutaneous membrane, and then you have the subcutaneous. I'm going to show you a picture of a cadaver. I want to demonstrate how thick this actually is. We're just looking at a diagram of my drawing, which isn't quite the scale. But I'm going to show you a video here. It's from Glands. This is the dorsal part of the hand. You can see that on this side, the lateral side of this person's hand, the cadaver's hand, the skin has been removed. They're grabbing at the skin, the cutaneous membrane. They're grabbing at the cutaneous membrane with tweezers. And you can see them tug on it and move it around. That's the thickness of the cutaneous membrane. It's not always the same thickness everywhere. And remember, if you go to the palm, it gets thicker. but that's how thick it looks here for thin skin. You can see the areolar connective tissue holding everything down. And then the yellowish stuff that's deeper, that's fat, the adipose, the subcutaneous. Again, you can see the thin skin found on the back of the hand. And then on the palm, where we have thick skin, it's thicker. Take a look at how they grab it with the tweezers. The thickness of the cutaneous membrane is thicker, and you can see the adipose deeper to it. I encourage you to look further at Ackland's. Obviously, it has some great videos showing the cadaver. The papillary layer, once again, has areolar connective tissue, has blood vessels. Therefore, it's going to be the one nourishing the epidermis. It's giving nutrients to stratum basale so it can undergo mitosis. The term for these bumps, like this bump right here, the term for this is a dermal papilla, or plural papillae. AE in Latin is pronounced E, and that's plural. If you're referring to one, it's a papilla. If you're referring to many, it's papillae. These are the ridges that you find in the dermis. They create texture in our skin, and when they're organized just so, they're the ones that create fingerprints. What's neat about them, or what can be found here besides blood vessels, you can also find a special sensory receptor. We've already talked about Merkel cells, but that's only found in stratum basale. Here we can find another receptor. It's called a Meissner's corpuscle. Meissner's is a person's name. You may also see it called tactile corpuscle. It's the same thing. I'm going to refer to it as a Meissner's corpuscle. Since it is also fairly superficial, but it's in the dermis, it does sense a lighter touch, but specifically light flutter and grip. Light flutter and grip. The Merkel cell was a single neuroepithelial cell. A Meissner's corpuscle is a little more complex than that. What a Meissner's corpuscle is, let's take a look at the histology down here. This funny looking thing is a Meissner's corpuscle. Whoops. Here too, this is a Meissner's corpuscle. What it actually is a nerve ending. So this is a sensory neuron. And what you're seeing, that oval shape with some texture, that's the Meissner's corpuscle, made of connective tissue, wrapped around the sensory neuron ending. Think about, let's say I were to poke myself with my hand, or with this pen onto my hand. My hand is the receptor. So I'm poking myself with this really sharp object. It hurts, it's pointy. But what if I wrap my hand with this scarf and then again poke myself with the pen? I still feel it, but I feel it differently. I don't feel that sharp point anymore. Now it's more of a dull thud. That's what this Meissner's corpuscle is doing. The connective tissue is changing the sensation that this sensory neuron is feeling, that it's sensing, that it's sensing. Meissner's corpuscles, therefore, aren't for just light touch or for pain or whatever. They are for light flutter and grip because of that connective tissue covering. And they're always found in those dermal papillae. They're always found in those dermal papillae. And therefore, they're found in the papillary layer within the areolar connective tissue. The reticular layer, once again, has dense, irregular connective tissue. It does not have reticular connective tissue. It has dense, irregular. The word reticular is referring to a network of stuff, collagen and elastic fibers. Here's histology staining for those different fibers, elastic fibers and collagen fibers. You can find more blood vessels, you can find more nerves, and we can start to find glands. We'll talk about glands here in a second. One other thing that you can find is another receptor type. This receptor is deeper, and it's also got a lot of connective tissue, way more than those myxor corpuscles, a lot of connective tissue wrapped around the nerve ending. These are called Pacinian corpuscles. Pacinian is a person's name, and the C sounds like an S, Pacinian. They're also called lamellated. The word lamellated means a bunch of concentric circles in layers. So you can see how it looks layered with all those circles. These Pacinian corpuscles sense deep pressure and vibration. Deep pressure and vibration. That's the key word there. Since it is so deep, this is showing these Pacinian corpuscles. Since so deep in the skin, you need more pressure to activate them. They're also very large compared to the others. Look at what's labeled here as a DPAP. These are dermal papillae. This is where you'd find myxor's corpuscles, and they're going to be really small. Pacinian corpuscles are much bigger. Pacinian corpuscles, by being deeper and by having many layers wrapped around the nerve endings, they sense deeper pressure and vibration. Their structure and location relate to their function. The layers and location, the deeper location, relate to the function of sensing deep pressure and vibration. We can add this to our diagram. In the papillary layer, we'll have to make some space, but let's add a nerve ending. And a myxor's corpuscle has an oval shape and then kind of squiggles around. So that's surrounding, let me do that again. It's surrounding the nerve ending. It's a myxor's corpuscle. A Pacinian corpuscle is going to be found deeper. Let's draw a nerve ending here. Pacinian corpuscle has many circular layers that cover the nerve ending. That again differs from Merkel cells. Remember, Merkel cells over here? Merkel cells are neuroepithelial cells that sense the light touch and then send that signal to a nerve ending. So they're not covering the nerve ending, they communicate with the nerve ending. In the dermis, I said that we can find glands. These glands tend to be deeper, so they're found usually in the reticular layer. One gland you can find are sebaceous glands. We briefly went over this before. If you recall, sebaceous glands release sebum. It's an oily secretion. It releases it through holocrine secretion. It's where the cell dies and releases its entire contents, holocrine secretion. The purpose of this oily secretion is to lubricate your skin and hair and also waterproof it. it's good to have a moderate amount of the sebum on your skin and hair. Too little, your skin and hair dry out. That's not good. It feels, your skin can get damaged very easily, and it can also hurt a lot. If you have too much, then you're too oily, and it can could not help with infections, and it could also lead to things like acne. So you need just a moderate amount. Sebaceous glands in moderate amounts inhibit bacterial growth, and like I just said, if you have too much oil, too much sebum, it could block your pores and lead to things like acne and other skin conditions. In this longitudinal section on the bottom left here, you can see skin. It's thin skin. We can see the stratum corneum, the flakiness of the stratum corneum. And here's the epidermis. The papillary layer is really thin. The reticular layer goes down to about here, roughly. It's not perfect, the border. And then you can see the subcutaneous down here. Sebaceous glands are always found next to hair follicles. This is a longitudinal section. We can see the hair follicles. They're really long when they're cut perfectly. They're not always cut perfectly. You can see some are cut cross-section, some are cut at a diagonal. There's still hair. And we'll go over how to identify hair more coming up. But next to those hair follicles, we can find these groupings of lighter colored cells that I'm circling in green. This is lower magnification on the left side and higher magnification on the right side. Look at these light colored cells. They're light colored because they're filled with sebum, that oily stuff. It reflects back at you a bit, and it secretes out to the canal, the hair canal, so that it can wick up the hair onto your skin. Sebaceous glands are found pretty deep, but also found next to hair, always found next to hair. Here is another view. I showed you a longitudinal section. Here is in cross section. Here is the hair and the canal of the hair, or where the hair would be. And then over on this side, you can see the sebaceous glands. They still look the same. Looks like a cluster of grapes, light colored. Those are sebaceous gland cells. They die, release stuff, and then the deeper ones undergo mitosis to produce more. Besides sebaceous glands, we also have sudoriferous glands. Remember these? There's two different types. Sudoriferous is just a fancy word for sweat. We have two types of sweat. When you think of sweat, you probably think of eccrine sudoriferous glands. This is by merocrine secretion. Merocrine means you're releasing by exocytosis. This is the watery secretion, the watery sweat, and it's found on most of your body, on most of the surface of your body. This helps with thermoregulation, maintaining your temperature. You sweat when you're hot, for instance. To identify eccrine sudoriferous glands, look for small lumens. These cluster of cells are close together. Compare that with apocrine sudoriferous. This is by apocrine secretion. Apocrine is where the top layer, the superficial layer, is shedding. And because of that, it's not so much a watery secretion, it's a thicker, has a bit more lipid secretion. It's thick, tends to smell. You find this in your armpits, your axillary region, and your pubic region, your genital region. The lumen is much larger as a result. By having a larger lumen, it can accommodate for this thicker, more viscous secretion. Compare that over here. Here's our apocrine glands. You can see the larger lumen, larger white space. They all look like a bunch of circles, a bunch of irregular circles, because what it really is, what these glands, how these glands are really shaped. If you take a look at this picture here in the middle, these glands are actually tubes that have just been coiled a bunch. If I take a wire, if you look at my screen, if I take a wire and look at it straight, but then bend it and twist it, now it's very convoluted. If I just slice it, you'll just see a bunch of cut pieces. That's what you're seeing here. All those circles are cut pieces of tubes that extend to the surface of your skin. And these are what secrete sweat to the surface of your skin. Eccrine pseudoriferous have narrow lumens. Apocrine pseudoriferous have very large lumens. Let's add this to our diagram. Let's make a little bit of space. I'm going to draw a hair in this region. So there's a piece of hair extending up. Next to the hair is where you'll find pseudoriferous glands. They always come in large bunches. And the cells are very light colored. Whoops. These are sebaceous glands. Note how I also drew this in the thin skin section. Sebaceous glands are not found in thick skin. Sebaceous glands are only found by hairs, and there's no hair on your palms or on the soles of your feet. Eccrine pseudoriferous Eccrine pseudoriferous has a narrow tube, a narrow lumen. These are simple cuboidal cells. So let me trace this in with a different color. Simple cuboidal cells surrounding a lumen. Or you know what, I'm gonna do it a different way. But these are simple cuboidal cells, narrow lumen. Apocrine pseudoriferous looks very similar to larger lumen. goes to the surface of the skin. I'm going to exaggerate the size of the lumen here. There is our apocrine pseudoriferous gland. All these cut circles are actually continuous with the tube that I've drawn. So please keep that in mind. It's all one piece. We'll continue this drawing, looking at more specifically at hair in the next video. But before I go, I just want to show you more resources of where you can look at the histology of this. If you go to our YouTube page, the Microscopes and Histology playlist, you can go to the skin histology video, Meissner's and Pacinian corpuscle histology, nail anatomy. There's plenty of videos here. You have Ackland's available to you. You even have complete anatomy. If you go to complete anatomy, click on complete anatomy, skin with hair, you can see a model that looks like this. And what's neat is you can click on things like here, I'm going to click on sebaceous gland, and it'll tell me all about it. And you can see it in 3 dimensions. You can see the different layers, the papillae. You can see Meissner's corpuscles right here. Call them tactile corpuscles, same thing. So there's a lot of resources out here. I don't want you to get too overwhelmed, but they're there if you need it. All right, so we'll continue talking about hair and nails and skin conditions in the 3rd and final video of this chapter 5. So I'll see you in the next video. Let me know if you have questions. Leave questions and comments in the discussion. Talk to y'all later.